And hello again. This is to continue the musculoskeletal system parts on muscles now. You've covered all the bones in the first 3 lessons. So moving over to the muscles. As always, here are the EDUQAS specification guidelines as to what we're going through in this bit. Mainly what is happening once you look inside a skeletal muscles. What have you got? Let's think of it as microanatomy, what is actually there in terms of proteins and protein filaments. How do they interact with one another on a biochemical level? Followed by a bit more of a generic thing on fast twitch and slow twitch fibers, and linking everything back to the use of biochemistry in the muscle, so creatine phosphate, using up glucose and glycogen stores, and that would be it. So key thing would be identifying the substructures. That basically means you need to be able to label both on diagrams as well as actually on histological pictures or microscopy pictures of the tissues that you would find. You need to know how they are functioning with one another. Therefore, the sliding filament theory, this is actually the big how does it work, the physiology. This is then how the anatomy, the bits that we've got, how do they function. So the sliding filament theory is going to be how do those muscle fibers actually manage to contract. Blood flow changes between fast twitch and slow twitch, mitochondrial density and so on, what makes one looking darker, one looking brighter, energy sources and muscle fatigue and cramps. Here we go, that's the lesson outlined. Keywords here for you, it's as always a bit of an onslaught of quite a lot of language. We'll get through this here nice and slowly. You should already know, either from your GCSE days or from when we did the ventilation of the lungs, that muscles work in pairs, a bit like the external and internal intercostal muscles in your rib cage to expand your rib cage up and out. They're working in pairs. In your arms, you've got the biceps and the triceps. In your upper leg, your thigh, you've got the hamstring and the quadriceps going forwards and backwards with the lower leg. You also should know that we have got voluntary and involuntary muscle groups. You know from the nervous system unit that you've got the somatic peripheral nervous system controlling the voluntary and the autonomic nervous system controlling the involuntary, whether that's now through the sympathetic or the parasympathetic branch. That's that. You should already have that as a background. You also, from your core concepts, know that you need a lot of mitochondria, not only for the contraction, because you will hear there where the ATP is needed in the contraction, but from the aspect of protein synthesis with regards to attaching the amino acid onto the tRNA. That's where ATP is required in the role of making the proteins. However, you will get to the power stroke. That's where a lot of ATP will be used as well. Muscles are attached to bones by tendons, ligaments, and bones. Muscles can fatigue. Now we will look at the fast and slow twitch fibers and which ones can actually resist to fatigue and why they can resist. And the physical function, this is actually going to be covered at a later point. The physics, the literally which angle which attachment moves a bone through which angle. That's a combination of joint and muscle function at that point. But this is part of it you should know, but part of it is an extension of what you already know. You then have this should be familiar from core concepts as well, that we have got 3 different types of muscle. We've got the 4 tissue types, one of them being the muscular. The other ones would have been your endothelial, your nervous, and your connective tissues. Now, muscular can be split into your 3, your cardiac, skeletal, and your smooth muscle. However, from here, you will need to be able to, from a histological picture, you've got in the bottom the 3 circles here, you've got images of what it would look like under a microscope. You will need to be able to identify which one is which by, it's mainly to do with are they striated and where are the nuclei. Have you got, we'll get to this just now. So for now, I'm actually thinking we have done some of this in the core concepts bit. So let's hope at least some of this will be there in your heads. The skeletal muscle is seen by its striated or stripy appearance. The stripiness of it we'll get to a bit later on. It's to do with how the individual proteins, the actin and the myosin, how they are arranged within each of those myofibrils. So we have got the arrangement of proteins which is a regular pattern, and therefore we get a stripy, striated visual appearance. Even though we can't see an individual protein, many of them will give us a visual difference. Smooth muscle is very often seen by having one nucleus per cell, and the cells are spindle shaped, some sort of a longish with narrow ends at the edges. Cardiac muscle is a little bit in between those two in appearance. It does a little bit of a dodgy thing with the nuclei, but not enough to be skeletal. In skeletal muscle, your nuclei are pushed out of a multi-mega cell, let's call it that. Let's do that properly when we've actually got the pictures coming up in a second. So nuclei, mitochondria, endoplasmic reticulum and fibers give muscle cells their individual appearances. So let's start with the skeletal muscle. A skeletal muscle cell is actually a cell where several individual cells are fused together in order to make a much larger feature. Idea of this is you can have longer protein contraptions that will then overall lead to a wider or bigger contraction overall. So what do you do when you fuse cells together? You have got the nuclei with all the information from all those cells. They then don't fit into the protein part of the cell, and they just get squished to the side as if they were no longer important. But you will see that in the next pictures coming up. The nuclei, it's a polynucleated thing, and the nuclei are on the side. You get a huge cytoplasm. It's then no longer called cytoplasm, it's called sarcoplasm. Literally that sarcomere, sarcoplasm, sarcoplasmic reticulum, everything with sarco has to do with the actual individual muscle contractile unit, let's call it that, to do with that. Now, why do you need loads of mitochondria? I think I said that earlier, both for the production of the protein as well as for the contraction and the power stroke you'll get to. The endoplasmic reticulum, from your core concepts, you should remember that that is the area that produces proteins, loads of ribosomes located on lamella. Now, if you've got lots and lots of endoplasmic reticulum space all added together from those various different cells, you then call it a sarcoplasmic reticulum. Just bigger, and in this case bigger is better. And the main fibers that you will need to know, the actin and the myosin, we will get to because they are pulling into one another. Here we go. This one is an appearance of a skeletal muscle. What I'd like you to look out for is that you've got the nuclei in lumps on the sides, are literally cluttered together, so the polynucleated thing. Here as well on the right hand side you can see that many nuclei seem to be within the same cell, and you've got them pushed to the side. Striated appearance, if you look closely you seem to have some sort of a stripy striated appearance. That is not actually where the actin and the myosin is, that's in a 90-degree angle towards it. It will become clear in a little bit. If you look at this here, what you're looking at now is actually the same physical orientation as the previous picture. So as if your entire muscle cell was running from left to right. Within bigger structures, you've got little structures that function. So within the kidney, you've got the nephron as the functional bit. Within your lungs, you've got the alveolus as your functional bit. And within a muscle fiber, you've got thinner stringy things of the myofibrils. We're going to get to the large to small organization in a bit. The smallest contractile unit is your sarcomere. Now your sarcomere is an arrangement of different proteins. The keywords you need for now is that we've got a thicker protein, myosin, and you've got a thinner protein, actin. If you need a memory aid for that, myosin's been eating loads of marshmallows and M&Ms and therefore it's a little bit thick, and actin has been eating loads of apples and therefore it's thin. So you've got a thick and a thin filament. Now they are arranged in a way that the actin is actually on a fixed end plate, the Z discs. If you look at them in a 2D version, then obviously they look like lines here. But if you look underneath, if you look at that as a circular arrangement, it's a Z disc. So they are circular areas where all the actin would be physically attached to. So the actin is attached. The myosin isn't really attached. It's kind of held in place a little so it doesn't fall out of place. But the myosin itself is floating loosely in between the actin. Now the contraction part will be happening where the myosin and the actin are close to one another. If you look at this now, you have got an area where the Z disc is. The Z disc itself, a little bit to the left and to the right of it, you don't have myosin. Myosin is a really thick protein. It's a thick filament. So if you stack many myosins all on top of each other, that will become a dark looking area. If you haven't got myosin, you've only got the thin actin looking area. You've got the light band, you've got the I band. Okay, so the A band is for a dark looking area and the I band is for the light looking area. This diagram may need to be printed out empty and for you to relabel. So the idea, the Z disc is your end where your actin is attached, myosin is in between, and you've got the different bands, the dark looking band and the light looking band. Now these bands will shrink into one another as we've got a contraction. If you look closely here, the A band actually halfway, the dark band, that dark band, the entire stretch of dark band, actually has got a little bit of a brighter area in between. The difference here is you have got myosin, but you don't have the overlap zone with actin. Look at the top, the squiggly bits where the actin is, the squiggly bits where the actin is as such. That squiggly bit has got an overlap zone with the myosin and an area where it's just actin by itself between the end of the myosin and the Z disc. Where you've got both actin and myosin, you've got really dark areas in the A band. And in the middle, you've got the H zone, which is darker than the I band, but it is still not quite as dark as the rest of the A band. So that is now very basic anatomy, or microanatomy if you want to call this, what you can actually figure out is there. This slide should help you orientate yourself, as in initially if you get a muscle, the muscle itself has got some connective tissue bits around it and in between, so the fascicles, and sometimes you've got even tougher collagen tissues on the side to hold the muscle in place. Within you've got individual muscle fibers. A muscle fiber itself is made up of myofibril. If you then look at a myofibril in detail, and if you were to cut that open and see what's inside, that is where you're going to find these individual sarcomeres. So the sarcomeres are inside a myofibril is inside a muscle fiber, and the muscle fibers altogether make up a skeletal muscle. This is just orientation. If you take different cross section through a muscle and then hold those underneath a microscope, you obviously see different things. If you were to have your cross section lengthwise, you would see the liney, stripy appearance. If you do the cross section in a cutting through the myofibril, then you'd always get sort of a dotty appearance, but that would not be seen under a light microscope anymore. The dotty appearance, that would be an electron scanning microscope. These ones, even though thick proteins, they would not be visible by naked eye. A bit of medical jargon: epi, peri and endomysium. So epi is always towards the outside, endo towards the inside, peri is the sort of in-between area. And if you're taking a muscle apart, this is now large scale again, the very outside is your epimysium. You have got in that, you have got individual muscle fibers within that. So literally just orientate yourself on this diagram. I have yet to see a question in an exam on this. I have not yet seen any labeling required. I've got a few pictures for you here in case it was to ask you to look at what is a fascicle, but I am yet to see any questions on labeling. There's not really anything other with regards to function, other than it just needs to all slide and be able to move past each other, and hence the connective tissue, also to hold it all together, obviously. Same diagram from earlier, just a little bit bigger to allow you to get a bit of an idea what's going on. What you can see here is the supply of blood vessels. What you cannot see is where the nervous system interacts with this whole thing. That is on a much smaller scale. It literally goes onto the sarcomere itself, but we'll get to that. We'll get to that in turn. So what I have named earlier, the fusing of cells, the fusing of cells and cells kind of mushing themselves together, and you end up with a polynucleated cell. I have used the word polynucleated, and you've actually met a polynucleated cell already when we did plant reproduction in component 2. You have met the egg cell, which does its first meiosis, then it loses 3, then mitosis, 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 to get 8 nuclei altogether. And that version of being polynucleated is from mitosis, so choanocystic. However, what we've got for the muscle cell is that actually those cells have started as individual cells, each with its own nucleus, and they've literally formed a syncytium. They're combined together, cell membranes fusing with each other and spilling their cytoplasm into one another, and also the nuclei therefore into one another. Just 2 different ways of being polynucleated. Luckily, in your syllabus, you do not need to know anything about the neuromuscular junction, so literally a synapse between a nerve impulse arriving and then the muscle itself picking up that signal. This diagram would kind of show you a little bit more as to how it works with regard to the T-tubule, and then opening various channels on the individual sarcomeres to allow the inflow of ions. But what you do need to know comes afterwards: what happens when those ions flow in. In a nutshell, the T-tubules you've seen on the diagram, they make up channels and canals where they're flowing their information to 3 sarcomeres at the same time, 3 sarcolemmas, sarcolemma being the cell membrane of those 3 muscle contractile units together. Idea is you're going to have the contraction at the same time, so you're sending information to several sarcomeres at the same time so that they would be contracting at the same time. I have actually at a later point in this video uploaded the link to a much better video, so I'm not going to spend too much time with this GIF here. This GIF is taken from a video where it's explained beautifully how the muscle contraction actually is happening. It's called power stroke. You have got in this diagram, you've got actin and myosin, you've got that little overlap zone, where actin and myosin are close together. So if over the T canals earlier from the outside you're getting an electric message arriving in the nervous system, you are opening up calcium channels. Think of this as being the action on the actual sarcolemma. So the cell membrane is initiating the inflow of calcium ions, so calcium channels open, calcium is flowing in. That calcium is attaching itself to the actin, that protein, that thinner filament. The actin at the moment hasn't got the binding sites for myosin heads, those little sticky out things from the myosin would potentially attach themselves to the actin, but they cannot attach themselves anywhere on the actin because you've got a tropomyosin blocking up the binding sites on actin. So the binding sites are potentially there, but you've got a chemical blocking them up, literally stuck in those binding sites. Now that tropomyosin is being moved and twisted as actin is being twisted out of the way. You're therefore now exposing the binding sites. The myosin heads can attach. The myosin heads, there is a power stroke cycle with ATP attaching, ATP splitting into ADP and inorganic phosphate, releasing one of those, releasing the other one of those. It's a nice little cycle which allows that little bit of a stretch, attach, pull, release, stretch, attach, pull, release. So that's literally a bit of a cycle which we'll look at separately. Again, I've uploaded really good videos for that, so me talking here is actually a little bit pointless. I'm moving on. Right, this diagram should not be unfamiliar. You know by now that the myosin is the thick filament in the middle. The actins are the thinner filaments, the thinner proteins. They're attached to the end lines, to the Z lines, Z as in end. The dark band is the A band and the light band is the I band. Okay, stupid memory aids, all of those things do help and do work. So why do we actually get that contraction? This is a different diagram, different set of diagram to show you the contraction of the actin actually being pulled into the myosin. You found that little GIF earlier, seeing that there's some little sticky out bits on the myosin that will attach themselves to the actin and pull and pull and pull and pull and pull. These diagrams here should give you a rough idea as to if you've got a relaxed muscle, the top diagram here, you've actually not got too much of an overlap zone between the actin and the myosin here. However, the more you contract, the more the whole thing is pushing into one another. Now, if you look at the microscopy images here, you can actually at some point no longer see that H band in the middle. The H band was the slightly lighter area within the dark area, within the A band, where the H band's here. That's kind of disappearing, and you're getting the scrunching together of the whole thing happening. So, keywords so far. You had a whole muscle has got inside it, muscle fibers has got inside it, the myofibrils. The myofibrils themselves are long tubular-like structures. Each of them has got back to back, a sarcomere, a sarcomere, a sarcomere, a sarcomere. The sarcomere is the smallest contractile unit. It's surrounded by the sarcoplasmatic reticulum. So the sarcoplasmatic reticulum is literally the endoplasmatic reticulum, which was your protein manufacture. The whole thing needs to have a whole lot of mitochondria to get the whole thing working. Active, it's an active process. You need oxygen. You need to have your ATP made for through aerobic respiration. The two protein molecules that you've got so far were actin and myosin. I have not yet been clear with the troponin and the tropomyosin. The troponin is where the calcium attaches to. The tropomyosin is the thing that is blocking the binding sites. Once the calcium is attaching itself to the actin twists, it's attaching on the troponin, and it's twisting the actin in a way that the tropomyosin is moving away from the binding sites so that the myosin heads can attach to it. The other video is so much better at explaining that than I am. The thick filaments are the myosin, the thin filaments are the actin. It's repeating units, and the smallest contractile unit is called your sarcomere, about 3 micrometers long. If you remember the sort of scales, if you've got an average nucleus, that's somewhere between 5 and 7 micrometers in size, similar to a bacteria. That's the sort of size of a prokaryote. And bigger plant cells can get up to 80-90 micrometers in length. The thick filaments, if you stack them on top of each other, make up the dark bands, the A bands. The thin filaments make up the pale and I bands. The Z lines are the end of each contractile unit, and the actin is attached to one of those. The H lines is in the middle of the A band, where you've got only myosin, so no more overlap zone, only myosin. And the M line itself is are the darker bands within the dark band, actually within the A band. Don't think I've had that one in a diagram yet. The dark band itself has MHM, so overlap zone of myosin and actin, then only myosin, and then myosin and actin again. This one is one of the 2 videos for you to actually really watch and get a focus on. At which stage do you attach the ATP? At which stage does that then change? Does it stretch the arm outwards? Does the actual splitting of ATP into ADP and in the organic phosphate, does that then stretch? Which bit does actually then do the pull part or the attach part? And when do you release each of them separately? The ADP has to be released again, and the ATP has to be released again. And all of that is kind of becoming a from there to there to there to there. This is a typical table drawing thing. So I'm expecting, as part of when we do the group work, that I want to have drawn on a table. I'm not going to reinvent the wheel. There is a fantastic video out there. You've got this link in the PowerPoint as such. I'm pretty sure that through YouTube you can't click on the screen right now. I would have to be clever enough to find a way of linking that. So there are good videos out there to explain that power stroke itself. Please watch them. It's in your hands, a poster, a table drawing, a flow chart, whichever way you focus or function your understanding of one of those larger processes. This is potentially part of a larger question. Now it's an option, so it won't be a 9-mark question, but you do have 4-mark and 6-mark questions in the options, which could be that explain how actually actin and myosin make a sarcomere contract. So that is one of the key things, one of that unit, and should be in your heads. Yet another video picked for you. Watch them, please watch those. Right. This one here is my basic version of what is actually causing the contraction. Please improve on this. Please improve on this. It's by no means complete. The action potential arrives. It comes in through the T tubules. You get depolarization, which causing calcium channels to open. So the calcium channels open. Calcium is a 2+. It flows in. It binds to the troponin, which is on the actin. So troponin is where the calcium is binding to. The actin itself is changing shape, which is twisting the tropomyosin out of the binding sites. The way I'm remembering the difference between troponin and tropomyosin sounds a bit weird. Tropomyosin sounds a bit like mayonnaise. And mayonnaise you can stick into binding sites to make them not work, and then you can push the mayonnaise out of the way and then it works again. I've got no idea why that works in my head, but I hope it works for you. We'll find something better, of course. So the tropomyosin is being twisted and moved out of the way so that actually those binding sites on the actin are exposed, and then the myosin heads, they're just little protein protrusions that they can actually attach to. It's called forming a cross bridge when those heads bind. So when the heads make contact and bind, that's when you've actually got the connection made. Okay, the myosin head at that point, as it binds, it's got still the ADP and the phosphate still attached from the previous stroke. The head is pulling back into its relaxed state at that point. So it rotates and pulls the actin past the myosin. That's when actually the pulling bit happens. Weird enough, at that point, you do not use up ATP. It's literally a pulling. The protein wants to be in its original place again. It wants to be sucked inwards. It wants to be pulled back inwards. At that point, we've got the release already of the ATP and the DP. The ATP binds, it breaks that cross bridge, and it stretches out the myosin head. So the ATP is actually being hydrolyzed in the stretching part rather than the pulling part, which is a bit counterintuitive. Please, that bit here, you will need to have more detail from your videos. At what stage exactly you have got the phosphate part released? At what part have you got the ADP part released again? Now that keeps going until the calcium ions have been pumped out again. The calcium ions don't want to go out by themselves, so they will have to be pumped out. So if you've got way too much calcium in your system and way too much calcium therefore flowing into your cells, that can lead to cramps. High calcium levels have actually been causes of heart attacks, of just contractions, literally involuntary contractions, too much calcium or too much calcium in the wrong place. The next picture is literally just for the sake of making this a bit more memorable. I did already in class at some point when I mentioned the idea of rigor mortis, and as mammals die, that you've then got a phase after death where the animal or the muscles are completely rigid. The picture's coming up now, so look away if you're not comfortable. What you can see is a dead cow, roadkill by the side of the road, all muscles stiffened up. Those calcium ions that were moved from the T area outside the sarcolemma into the cell, that needs to be pumped out again. This is an active transport thing, so you'll need your mitochondria active, you need aerobic respiration and your ATP made. As an organism dies, the ATP is running short and running out, so the pumping outwards no longer works, and the calcium inflow still works. So you get contraction, and it just takes several hours for that whole thing to be released again.